Okay. Hi, I'm Beth Moorfield from Nature's Structural and Molecular Biology, and I'm here today speaking with Steve Kay from the University of Southern California. It's nice to see you again, Steve, and let me move this box so I can see you. There we go. All right. The talk that you gave a few days ago was on the role of the circadian clock in cancer pathogenesis and the development of cryptochrome stabilizers as anti-cancer therapeutics. And I wanted to ask you in your introduction, I think most people are familiar with the concept of disruption of the circadian cycle is associated with increased cancer risk. But you, in your introduction, brought up some evidence that, in fact, clock BML can actually work as a function as an oncogenic driver in some cancer cell types. And could you just give a brief introduction for our audience now, what the evidence is?
Yeah, absolutely. And thanks so much for your interest in this. Look, a lot of really great work, primarily really over the last 10 years, has made a number of what I would view as systems level connections between the circadian clock and the cancer process, shall we say, oncogenicity or the sensitivity to develop particular types of cancers. And so there's been a broad framework that circadian function seems to be linked in genetically to clock function or dysfunction. I think the challenge there has been, how do we convert those kind of systems genetics approaches, including epidemiological work, which, for example, some investigators have argued there's a signal for breast cancer risk factor from Schiff work. So how do we take all that data in sum and try to develop a framework for how we might think the circadian clock and its individual proteins are targetable with respect to cancer? There's definitely been a general view that in a lot of tumors, if you use molecular probes and you go in and look at circadian regulated gene expression of the core clock, the core clock seems to be often disrupted in certain types of tumors. But if we combine the sum of mouse genetics and human genetics, then sometimes a particular clock gene seems to be a risk factor for a cancer, and other times in another cancer, that same clock gene can appear to be acting as a tumor suppressor. So, what we need is a more systematic way of looking at clock gene function in a cell-specific way in individual cancers. I don't yet see anything emerging that's going to be a grand unifying theory about the circadian clock and cancer. I don't subscribe to the idea that the circadian clock itself is tumor suppressive or rhythmicity is tumor suppressive. I think we're going to have to do mechanistic proof of concept studies in individual cancers. So, we can take that as a framework, and then we have to sprinkle in serendipity. Mm-hmm. So, for example, what we've seen in a couple of different cases is we've been approached by cancer researchers because they've begun to find, direct molecular links between specific clock proteins and specific types of cancer cells. And this was very much the case with the work we're doing together with Jeremy Rich, who's at UPenn Medical Center, where Jeremy's group began to get some data. So some of it, for example, is like the TCGA database, where they came to me and said, look, this is pretty interesting. When we look at these so-called negative elements of your clock, like cryptochrome, high cryptochrome is prognostic of better patient survival. And conversely, when we look at the opposite, the positive arm of your clock, then we see that, for example, high BML expression is prognostic for poor survival or higher grade glioma. And so we started to investigate this together with Jeremy. And the first place that we started was simply to knock out clock and B-mel transiently in these glioblastoma stem cells that Jeremy focuses on. And we just got this striking result that I think neither of us believed, for the first 6 months. And the poor postdocs had to get back to the bench and sort of continually repeat in different formats. And that was that these cancer stem cells were uniquely sensitive to the knockout of clock or B-mel. And clock now means the gene or protein and not the general clock. Joe Takahashi's gene. So, if we knock out either clock or B-mel, there is an incredibly detrimental effect on survival of these cancer stem cells. And if we use a bunch of control cells, but most notably, other neuronal stem cell lines that have been derived from patients in the same way that the GSCs are derived surgically from patients, there wasn't any effect of clock and B-mel knockdown. And of course, for those of us working in this field for years, we haven't really noticed that if we were knocking down clock and B-mel to measure rhythmicity, that we were having any effect at all on cell viability. So this seems to be a fairly specific case. And, because this was published near the end of 2019 and then was confirmed and extended in a follow-up paper by Ron DePino's group, just to quickly summarize, what we found there is it's actually the epigenetic landscape of these cancer stem cells has been altered by a $64 million question process. So what is upstream that causes that epigenetic shift? But we could detect all of this open chromatin right over the sites where clock and B-mel normally are excluded from binding in cancer stem cells, but are allowed to ectopically bind in this case. And so what do you think clock and B-mel do when they're let loose like this? They do what they do in differentiated tissues like skeletal muscle or liver. They drive proliferative processes, they ramp up metabolism, they contribute to stemness. And this, I think, is where we should start to think of the real framework for trying to understand the clock in cancer is, what are those hallmarks of cancer pathways, the canonical ones described by Weinberg and colleagues over the years and are updated regularly? If you start looking at those cancer hallmark processes, then given the fact that the circadian clock regulates, 30 to 60% of the transcriptome in a given cell type, it shouldn't really come as a surprise that some of those key steps in those cancer hallmark pathways are themselves clock regulated. So that was one case where we went on to show that in PDX xenograft animal models, that genetically, if we knock down clock and B-mel before implanting those tumor initiating cells into mice, it greatly extended survival of the mice. So that was really nice confirmation that this was a significant pathway that should be targetable. The real challenge then comes from, how do we move into the world of, therapeutics and convert this to real therapeutic information? And again, here comes a sprinkle of serendipity on top of good sense, right? And I'd probably most days of the week have the former on my side. And that is, our lab had been doing phenotypic driven chemical screens for years. We're not the only lab that does this. Several other labs have been successful at identifying molecules that alter clock parameters like amplitude or periodicity, etc., period length, and then deconvolving their targets. And in that way, quite a few years ago, we published a kind of a step change type science paper where we showed that some of these molecules that we were discovering that were affecting the clock were actually binding to the flavin binding pocket that's conserved between cryptochromes and photolyases. And so we use those molecules for quite a few years as academic fun molecules, and published a whole series of sort of chemical biology papers, asking questions like, can we make this go in both directions? So can we either stabilize cryptochrome or destabilize it? Because these molecules have a very unique mechanism of action. They fit into the pocket of cryptochrome, and they displace this part of the ubiquitin ligase that docks into that pocket. So by displacing the E3 ligase, you raise the levels of the protein and stabilize it. So they're agonists, cryptochrome agonists. And we had a lot of fun asking, could we switch them in different directions? Can we begin to understand cryptochrome 1 versus cryptochrome 2 selectivity? And use these molecules to really probe cryptochrome function, all the way up to sort of more recent work that I've been less involved with my close Japanese collaborators, where they've made photoreversible forms of these molecules. So you can actually now have a circadian photo switch.
Nice.
Where you can kind of, stabilize or destabilize cryptochrome in a classic, optogenetics type format. So that's all great, and they're really fun tool molecules. So the question is, could they be therapeutics? Well, that's an expensive question to answer. So, COI declared here, we spun out a company that was willing to fund a parallel development of molecules that, weren't about having chemical biology fun and sort of, using them as real discovery tools, but really seeing if we could get a development candidate, a cryptochrome stabilizer that passes muster and is ready for a phase 1 clinical trial. And that's what Synchronicity Pharma has done. So we now, thanks to them, we have tool versions, so kind of earlier versions of their molecule that we show we could put in vivo, and they will extend survival in a glioblastoma PDX xenograft model. And at the same time, Synchronicity has taken this a bit further and actually developed a molecule that was approved for by the FDA for a phase 1 safety study. And so they've done a multi-dose escalated phase 1 study and shown that the two really key things, that even at high doses, the molecules are safe. At the highest doses, we do see a little bit of disruption of locomotor activity, but there's not a complete shutdown of the circadian system. And thirdly, thanks to all the work that so many of the speakers at this meeting have done, we can use gene expression as a biomarker for these drugs binding their target. So when we stabilize cryptochrome, there are specific changes in EBOX dependent transcription, and we're going to develop, if you like, an algorithm or a constellation of markers. But we showed we could do this in white blood cells from patients in the phase 1 study who were getting our drug. So this is, drug discovery is always great until it isn't. There's a long path ahead. These things can die at any point. But what we're really hopeful for is that this is kind of just the tip of the iceberg, and we can start to use this paradigm, and if you like, scan cancer viability space to actually look for those cancers that might also be sensitive to clock and BMAL abrogation.
I'm laughing because you're anticipating all of the questions.
This is great.
No, it's good. It's good. This is what I was interested in.
But it really, what I love about this story, of course, is the potential to treat patients who have a devastating disease. That would be an amazing blessing and privilege. But prior to that, though, it really speaks to, how all of this comes together. A neuro-oncologist who knew nothing about the clock, but was beginning to see signals of clock gene activity in their data. A chemical biology effort that's phenotype driven. all those years ago, you wouldn't have looked at the theoretical because there was no
Right.
crystals. I know, of course, we got to mention the word structure when we're talking today. But, there was no crystal structures of cryptochrome then, and you couldn't have really, created these molecules, but they came from a phenotypic screen.
Right.
And we have other molecules that have come out of that phenotypic screen that are also showing interesting potential as cancer therapeutics.
So you was also, so the drug that's actually in the phase 1 trial is not cryptochrome isoform specific, right? It binds both Cry1 and Cry2.
Oh, yeah. Well, you're asking, of course, a really great question. So what we haven't told the world yet, but we have the data that supports their Cry2 selective. So they have a much stronger preference for Cry2 over Cry1. There's no absolute specificity yet. Our academic studies developing tool molecules that are essentially fortuitously Cry1 versus Cry2. We can now, if you like, hack those molecules to try to understand, well, if they're fortuitously Cry1 or Cry2 specific, why? And what are the key residue molecule interactions that are driving that? So, we've got some great tool molecules to learn about specificity. And ultimately, it may be that we're going to have to, really knuckle down on that cryptochrome specificity, very likely, in order to truly have a successful drug in the clinic.
Speaking of structures, not that every part of it is particularly ordered, though, right? There are a few disordered regions.
Yeah, cryptochrome has some fuzzy disordered parts, but the nice thing is the pointy end of the stick that we're interested in, which is the FAD, the evolutionary concerned FAD binding pocket. And my lab's weird in that we've worked on cryptochrome and Arabidopsis in flies as well as in mammals, as have other people too. But yeah, that nice conserved pocket is definitely where the action is from a structural point of view.
Nice. So I mean, basically, if you're describing the changes in the epigenetic landscape changes, then based on this, I mean, it's nice to see like some mechanism in the background. That you wouldn't necessarily expect that the lethal effects on cells, right, would actually be always coupled to changes in circadian periods.
That's right. And so, I think in a way we have to be very careful about what rhythmicity tells us about a cancer cell. And in fact, when our paper was reviewed, we for sure had a very brilliant circadian reviewer, because they really held our feet to the fire on these, kind of noisy rhythms that we could detect in the glioblastoma stem cells. But I think our point is, well, look, this is an ectopic clock, but there's still some feedback going that can drive some rhythmicity. But is that remaining rhythmicity really relevant to what we want to do? Is it even a biomarker for the types of cells we want to target? I'm really not too sure that it is. I think probably the most efficient tool we have right now is probably scanning, and rather than, cells that have been in culture for 25 years, but, we're really interested, because of all the changes in sort of clinical sample collection and biorepository and 3D cultures. We're really excited about collaborations where, freshly isolated cells from patients can be put into spheroid or 3D cultures, and then we can test our drugs on those almost in real time.
Oh, nice.
And we're just standing on the shoulders of giants who have set up all of that. And I think, scanning cell viability using, genetic tools could be the most efficient way to do that. We shouldn't ignore the possibility that there's also going to be a tumor microenvironment effect, TME. So, we showed, I think, pretty nicely in glioblastoma stem cells, this cell autonomous effect in those stem cells of the clock, metabolism, stemness, proliferation potential. But what DePino's lab did was not only show that, nicely, because it's always good to see your stuff reproduced, right? That they could reproduce all of those cell autonomous effects of the clock, but they actually showed that there is a clock-driven chemoattracting molecule that's actually a direct target of clock and BMAL.
Oh, wow.
So that chemoattractant expression is ramped up in glioblastoma stem cells for the same reason all the other clock programs are.
Right.
What that chemoattractant does is it attracts these myeloid cell line microglia, which are tumor suppressive. And so, this is where, you have to rely on colleagues to just be very generous when you're naive about a complete area of biology, as we were. So we've had to really go back to school and learn a lot about these myeloid-derived suppressor cells. And they're important in most types of tumors, and including GBM, where they migrate into the tumor microenvironment and they play a tumor suppressive role there. And very often, for example, you can go into a tumor and use FACS analysis of the tumor tissue, and you'll see, as you use markers that identify these myeloid-derived suppressor cells, you see a reduction in Tregs, CD8, CD4 T cells, etc. So the clock is having a role in tumor suppression, and it goes in the right direction, because what they showed is if you abrogate clock by knocking down clock in GSCs, something that we hadn't really paid attention to, which is the effect on migration of these suppressive cells, will be an additional benefit. So, this is going to be very dynamic, and it's going to crop up, I hope, from many other researchers. So, several years ago, up at the Broad, there was a group that wanted to do a systems biology analysis of acute myeloid leukemia cells. So Ben Ebert lab. And they found that the key driver in those leukemia stem cells was clock and BMAL. And if you knock clock and BMAL down, you kill the leukemia stem cells, but not normal hematopoietic stem cells. So there's a parallel story by a completely independent lab, pointing perhaps to a similar process going on in AML.
This is really nice. They have I did want to ask before I move on, I was like, so so the cells are all dying by apoptosis, is that right?
Yeah, well, you kind of, you knock down clock and BMAL, and then the race is on to phenotype them in this dying cell population.
Right.
So the kinetics of all of this, I think, is something we need to explore in finer detail to fully answer your question. But we do see very clearly two clear signals in deep phenotyping. We see cell cycle arrest at the G2M boundary, and we see a dramatic increase in apoptosis when you use classical markers in FACS analysis. So, that appears to be the two key mechanisms we think that are driving the dependence, if you like, of BMAL and clock on cell viability.
Well, those reversible Cry2 inhibitors might come in handy for some of these.
Absolutely, yes, because in that way, yes, we can try to start to establish sort of an order of events when you knock down clock and BMAL with a chemical tool. Absolutely, spot on. Yeah,
that's going to be great. They have I'm going to ask like this for there are effects on, I know there are effects that of clock function on DNA damage response pathways, another very active area of investigation for new cancer specific therapeutic targets. Have you seen any effects on these, or are you investigating any of that? Or looking at overall DNA damage accumulation?
No, the absolute honest answer to that is we totally agree that should be a fruitful area, but we're just swamped at the moment. So, I think DDR is absolutely a place to look. And, at the meeting, Aziz Sankar and Katya Lamia both touched on that in their talks. And, this could also be interesting, right, in terms of thinking about what will probably be the first way in which a clock drug will be used in cancer, which is in combination of standard of care. So in GBM, for example, the way that patients are treated is, generally speaking, the first line of treatment is surgical resection. That's how we get these samples from our clinical collaborators. And then that's followed by a period of chemoradiation. And the chemo agent is temozolomide. And that's, of course, an alkylating agent. And the effectiveness of temozolomide is somewhat dependent upon an enzyme that reverses the alkylation process called MGMT. So it could be that if there are unknown actors right now that are also clock effective, that are part of the DDR, then we might want to start to look to see if a clock drug could actually potentiate a chemo agent or chemoradiation treatment strategy. So we've actually got some collaborators up at Harvard Medical School at Brigham Women's, Khalid Shah and Hiro Wakamoto. And in the, they're working with GBM patients who have different levels of responsivity to TMZ. So, more sensitive, more resistance. They've established cell lines of glioblastoma stem cells from those patients that have these different levels of TMZ sensitivity. And so now we can go in and probe those with our clock drugs on their own and in combination with TMZ. And again, it's amazing how collaborators have sort of opened their minds and their doors and their freezers to helping us, test these ideas.
No, it's really great though to see a lot of the small molecule modulators, the moving from research tools to therapy, and that there seems to be this wave, of
and I think, exactly. You're dead right, and the two key axes for progress is what other cancers might this work in, and so currently our lab is sharply focused on 4 cancers where we think this could be really relevant. GBM, colorectal cancer, where we've got an amazing collaborative team at USC. Again, they came to us because they'd seen a signal for clock genes in GWAS studies of phase 3 trials for colorectal cancer drugs. Again, just coming to us, right? Phenotypic driven. We think AML is there, and the Broad group don't seem like they're really following up on that. So we're starting to pick up on their story and sort of build that out in acute myeloid leukemia. But we've also seen this in liver cancer, specifically hepatocellular carcinoma, where we see that same sensitivity to clock and BML knockdown. So that's really going to be enough for a group that is not has no history whatsoever in cancer biology, working on the clock in 4 different cancers. What we're really hoping is, because of abstracts at ASCO and AACR, et cetera, we're really going to see a rise in this. And in fact, if you look at PubMed and you search for circadian and cancer, then after about 2002, there's just this steep climb. So I think last year in 2020, there were 350 papers that show up when you search for circadian and cancer.
Yeah, it used to be just shift work and jet lag and these guys, right?
Yeah, and those epidemiological studies have a role, right? Because that's the world of preventive medicine. So can we learn circadian hygiene? And can we have a preventive medicine aspect to circadian biology and how it relates to health? So Chuck Seisler telling us all to keep off our iPads at night. Even though there's a big biostatistical argument going on the robustness of the breast cancer signal, it's clearly worth investigating further. I mean, this is a lot of people that could be affected by it. But what nobody argues about is the metabolic disease that you see associated with chronic shift work.
Yeah.
I mean, I think there are 3 ways to do this. Sachin Panda posed this in a review a few years ago. You can You can basically, have circadian lifestyle, which is preventative medicine. You could have clocking the drugs, which is John Hoganish and his whole new world of how to look at cyclic drug targets and taking a single tiny skin punch from a patient and deconvolving that into what their actual circadian gene expression is just from a single time point.
Wow.
And then I think, drugging the clock, which is what we and others are into, where, direct modulation of circadian proteins in specific cancers could be a way of providing a patient benefit.
Well, no, it's really, really fascinating. It's been really, really exciting to see these develop. And really, really well. It's also nice to see like how much working out the mechanism using these has really informed where the therapies are going. And also it'll be interesting to see what it's not only for cancer but then other metabolic diseases, as you brought up from the drug. But yeah, so I think we might be out of time, but thank you so much for this. It's like,
well, thanks for your interest, Beth. You've always been a great supporter of our field, and we really appreciate it.
Yeah, that's great. Yeah, well, it was good. I hope to see you again face to face at some point soon.
Yeah, this room is getting pretty tiring. But it looks like it's on its way. Here in California, June 15th tomorrow is a big step change, and my lab people have suffered terribly, like everybody's in the last year, but now we're beginning to pick up speed again.
Okay, so not everybody is back yet. So
they are finally pretty much back, but we were at one point for a long time. We had to have 3 8-hour shifts to keep the density within state guidelines, LA County guidelines.
Yeah, I was noticing that. Yeah, it was pretty hard hit. I had my eye on that, wondering what was happening in the labs down there.
But fortunately, in our whole research center, we've had no active transmission. So
excellent. Okay. That's excellent news. Yeah. Well, as we're getting vaccinated, we'll slowly get back there. But
absolutely. Yeah. Well, wonderful to speak with you. Thanks for those great questions. Too. It makes me want to jump onto the bench and work even harder.
Okay. Well, good luck. I'll be in touch again at some point. Bye.
Thanks a lot, Beth. Bye-bye now.